Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on earth, and that's biology. So welcome to my new beginning section. So today we're going to talk about pollination, part of a class 12 chapter that is sexual reproduction in flowering plants. So we have been talking about class 12 on an alternate basis. So that is every Monday, Wednesday and Friday we talk about class 12 topic. Tuesday, Thursday, Saturday we talk about a class 11 topic. So today being Friday, we're talking about a class 12 chapter. We have started with sexual reproduction in flowering plants. So we'll continue with pollination. So we're done talking about how your stamen gives rise to anther and filament, and how anther produces pollen grains, and how the pollen grains become ready for the process of sexual reproduction. So then on Wednesday's class, we spoke about megasporangium, that is the female reproductive part gynoecium, which is represented by pistil or the stamen, and how it produces the female gametophyte, that is the embryo sac, right? So now that the male and the female gamete are ready, now how are they going to meet each other? Because what if they are extremely apart? So sometimes if they have to move from one plant to the other plant, so how are they going to move? So the pollen grains do not have legs to walk and find their partner, right? So someone has to help them in moving from their place to the place of the female embryo. So that process is known as pollination. So who helps in pollination? So we have pollinating agents like wind, insects, birds, etc. So that's all today's story. So we're going to talk about the process of pollination. So we're going to talk about how or what are the different types of pollination. Who are the pollinating agents? In different types of pollinating agents, what are the different characteristics of the plant or the flower? And then if a plant wants to stop self-pollination on its own, and it has to promote cross-pollination on its own, so how it's going to do. So that is the next step we are going to talk. So we're going to talk about in 3 parts. So we'll finish off. So that is all for today's lecture. So that is the entire story. So let's move forward. Pollination. So far we have been talking about the structure of the pollen grain and the embryo sac. So let's move forward. So I'll give you quickly a recap so that it's easy for you to understand. So we have been talking about your andrecium. So I hope you remember the andrecium has a structure like this, that is stamen. The stamen has 2 parts. One is anther and one is filament. Right? So this anther is bilobed, but it has a thecal wall like this. So when I cut it open like this, so it is present in 4 discrete corner-like structures. So this is known as tetrasporangiate because there are 4 corners, and inside each of the corners you have microsporangia. So microsporangia is the pollen sac. So this is the pollen sac. So this is surrounded by what is known as microsporangium. So this microsporangium has 4 wall layers. So what are the 4 wall layers of the microsporangium? So you have the epidermis, endothecium, middle layer and the tapetum. So the 4 wall layers are, the first one is first one is epidermis. Then you have endothecium. So then you have middle layer. And then you have tapetum. So these are the 4 layers of your microsporangium. So your epidermis is the outermost layer. Right, so your epidermis is the outermost layer, and the first 3 layers have the same function, that is, it helps in protection and it also helps in the dehiscence of anther, so to release the pollen grains. So these are the functions. Talking about tapetum, so tapetum is the innermost layer. So this innermost layer helps in the nourishment of the pollen grain. Plus it produces Ewbish bodies. So this Ewbish bodies help in the formation of sporopollenin, and then it also helps in the production of an enzyme called callase that helps in the dehiscence process. So in the center of your microsporangia, you have a group of homogeneous tissue. So this tissue is known as the sporogenous tissue. So this sporogenous tissue consists of the spore mother cell or the pollen mother cell. You can call it as the pollen mother cell, or it is called as the microspore mother cell, which undergoes the process of meiosis to form microspores. So you can call it as pollen grains, or you have 4 microspores. So it is 4 pollen grains or 4 microspores. So you can also call it as microspore tetrad, right? So this process by which the pollen mother cell undergoes meiotic division to form the pollen grains, so this process is known as microsporogenesis. Fine. Hi Rohan, yes, a very happy good evening. So thank you so much for joining my lecture. How are you doing? So did you get any updates, because you didn't update me after that? Yes, did you get any updates? Thank you so much Rohan for joining my lecture today. So please do like, share and subscribe. Right, so let's move forward. So this is microsporogenesis. Now each of these microspore develops into a potent pollen grain. So one each, so we talk about one pollen grain, so the fate of other pollen grains are the same. So one pollen grain further undergoes mitotic division and it forms one generative cell plus it forms one vegetative cell. So in majority of the cases, the pollen grain is released at this stage. So this is the 2-cell stage. Fine. And further in certain angiosperms, this generative cell further divides to form 2 male gametes. So it becomes the 3-cell stage division. So this is the 3-cell stage division. So then we went on to talk about the structure of pollen grain. 20,000. So you still have to pay 20k. Alright, so you take a decision. So you let us know what you are willing to do. So the support team asked you to pay so much. Fine. Okay, so I will also talk to the support team on my behalf. Okay, fine. Alright, Rohan. So you take a decision and let me know whatever. Anything is fine. So I have no hard thoughts. So if you were interested, so I thought I would help you, but otherwise it's completely fine. So you take a call and let me know. All right, Rohan? Yes. So this is the 2-cell stage. So talking about the structure of the pollen grain. So the structure of the pollen grain is that it has a 2-wall layered. So it has an outer layer. So the outer layer is known as XII and the inner layer is known as intine. So the XII layer is made up of the hardest substance, that is sporopollenin. So this sporopollenin is very resistant to temperature, pH, alkaline, etc. So this gives the appearance of pollen grains as a living fossil. At certain junctions on your XII layer, you also have germ pore apparatus. So these germ pores are the regions where your sporopollenin is absent. So the inner layer is made up of cellulose and pectin, and it is a very thin and a continuous layer. So this is the structure of the pollen grain. So moving further, we spoke about the viability of pollen grain. So what is the viability of pollen grain? It varies from one plant to the other, and pollen grains also act as potent allergens, but they can also be used as supplements to help in better performance, right? So further, your pollen grains can be preserved. So that method is cryopreservation. So how can you preserve the pollen grain? In a liquid nitrogen, that is minus 196 degrees, right? So this is how you preserve the pollen grains. Price is high. Okay, so I can understand. So I'll get back to you in the Telegram group. In the Telegram message, we'll discuss. All right, Rohan? Yes, I will also talk to the support team. Right. So moving further, so that was about the male gametophyte. So the male gametophyte was the pollen grain and we spoke about it. So now we're going to talk about the female gametophyte. So we are going to talk about the embryo sac. So how is it described? So we are talking about the structure of the pistil or the carpel. So we have the style. So this is stigma. We have style and we have the ovary. So the space inside the ovary is called as the ovarian cavity. So this ovarian cavity is also known as locule. So inside the locule you have what is known as placenta, a cushion-shaped structure. So this cushion-shaped structure is placenta, and to the placenta are attached the ovules. So ovules are the megasporangium, right? Fine. So megasporangia will be the embryo sac, or embryo sac is the female gametophyte. So now moving further, so you have the placenta and you have the ovule. So the ovule is attached to the placenta by a stalk that is known as funicle. So this attachment is known as funicle. So this portion where the funicle attaches with the ovule, so that is known as hilum. Fine. Alright, so I understand your problem. So I will get back to you. So I will talk to the support team as well. I will get back to you in Telegram group. Alright, Rohan? Yes, don't worry. Fine. So this is hilum and this is funicle. Fine. So now your ovule is surrounded by one or two layers, protective layers like this. So this protective layer is known as integuments. So this is your outer integument. So this is your inner integument. So this is protective layer. So if it is one outer layer, it is unitegument. If it is two outer layers or two integuments, it is bitegument. So innermost layer to the integument is nucellus. So if you see, the integument does not cover the ovule at one point. So this end is called the micropyle end. And exactly opposite to the micropyle end, you have the chelacal end. So this is nucellus. So that is fine, Rohan. So the pleasure is mine. So inside the nucellus, you have cells known as the megaspore mother cell. So this megaspore mother cell again undergoes meiosis to form 4 megaspores. So but out of this 4 megaspores, only one is functional. The others degenerate very quickly. Fine. So this process of the megaspore mother cell undergoing meiosis to form megaspores, so this is known as megasporogenesis. Fine. Right. So now this one functional megaspore will undergo meiotic division to form 2 nucleate stage, which will further undergo next meiotic division to form 4 nucleate stage. So this will further undergo meiotic division to form 8 nucleate stage. Right? So now this 8 nucleate stage is going to form your embryo sac. So out of this 8 cells, 6 of them forms the cell wall around themselves. Right? So these 3 cells are called as antipodal cells, and this end is known as your chelacal end. So this end is your micropyle end, and here one of them is your egg, that is the female gamete. The other 2 cells are known as your synergids. And egg plus the synergid is known as the egg apparatus. So this is known as the secondary polar nuclei. So this is the functional embryo sac. Thus your embryo sac is 7-celled and 8 nucleate. So your embryo sac is the female gametophyte. Alright, so I hope now the female portion is clear. So now we move to the part of pollination. So pollination is the process of bringing the male gamete, that is the pollen grain, to the female gamete, because they cannot walk on their own, they don't have legs. So suppose in case of papaya, there are two different plants. The male plant is different, the female plant is different, right? So in that case, how does pollination happen? So how does it move? So that process is known as pollination. And what are the different types of pollination? So we are going to talk about that. So the first type of pollination, so depending on the source of pollination, so first one is autogamy, the second one is geitonogamy, and the third one is xenogamy. So autogamy, so gamy is fusion. So everywhere gamy means fusion. So auto is self. So when self-pollination happens, that is autogamy. So autogamy happens within the same flower. So when one flower has both androecium and gynoecium, so then self-pollination happens, and that is known as autogamy. So the next one is geitonogamy. So the geitonogamy happens within the same plant but two different flowers. So this is within the same plant but two different flowers. Right? So one plant can have a separate male flower, can have a separate female flower, and when pollination has to happen between them, that is your geitonogamy. So xenogamy is cross pollination. So when it happens between two different plants. So this is cross pollination. Fine. So these are the three types of pollination that we have. So first is autogamy, the second one is geitonogamy, and the third one is xenogamy. Alright? Yes. So now we move forward and we'll talk about autogamy. So pollination within the same flower is autogamy. So transfer of pollen grains to the stigma of the same flower. So it is happening inside the same flower. So the normal flowers open and they expose the anthers and stigma. So complete autogamy is rather rare. So synchrony, the pollen grains and the stigma receptivity. So in case of autogamy, so what are the characteristics of the plant? So that is what I am discussing. Suppose if we have a plant that is doing autogamy, that is self-pollination. So what are the criteria or the characteristics that should be considered, or that is characteristic of a plant that is undergoing self-pollination? So in self-pollination, so it is happening within the same flower. So now I have a flower, and in this flower I have the androecium and the gynoecium. So the pollen grain and the stigma, so the pollen grain is ready, the pollen grain is going to come out, and the stigma should be able to receive the pollen grain, right? So the stigma should also be receptive enough, it should also be mature enough, right? So that receptivity should be there. So both the pollen grain and the stigma should be receptive together. So there should be a sync, there should be coordination between both, right? Sometimes when the flowers open up fully, there are also chances of cross-pollination. That is what is written in the third line. So anther and stigma should lie close together because it is self-pollination, there is no pollinating agent here. Right? So that is about autogamy. So these are the points that you should remember. Ma'am, update cup. Okay, so after class I'll get back to you. Okay, Rohan? Yes. So remember, for autogamy, so the stigma and the anther should lie close together, and both of them should mature at the same time. That is, there should be synchrony between the male portion and the female portion. But when the flower is completely exposed to the outer environment, there are chances of cross-pollination as well. Right? So complete autogamy will be rare in the case when the flower is open so much. So autogamy is seen, so examples are very important. So autogamy is seen in Viola, that is also known as common paxi. So in Oxalis and Camellia, so these are the examples. They produce, so Viola, common paxi, so autogamy is seen. So Oxalis and Camellia, they also show your autogamy, but they have two types of flowers. So what are the two types of flowers? One is chasumogamous flower, the other one is cleistogamous flower. So this chasmogamous is very, very charismatic. So it opens up so wide. So the petals move out so wide, the anthers are very, very open, exposed, the stigma is very exposed. Now there are chances of cross-pollination as well, right? So those type of flowers are chasmogamous flower. So cleistogamous flower is the flower which does not open at all. It is always in the bud stage, and in the bud stage itself, the pollination will happen. So actually the bud stage, the flower is not developed, ma'am, then how can you see pollination can happen? It is just that the flower does not open up, but the stamen and the stigma become receptive, or they become mature at the same time. So they do not open at all, so there is no chance of cross-pollination in your cleistogamous flowers, right? So in cleistogamous, anthers and stigma lie close to each other. The anthers are dehisced in the flower bud, transferred to the stigma. So everything happens in the bud stage itself, right? So this is invariably autogamous. So whatever you do, cleistogamous flowers cannot take cross-pollination. So what has to happen is only your self-pollination, right? So there's no chance of cross-pollen landing on the stigma. So this ensures a short seed set. So without the agents, pollinating agents, without any external source, so it is going to give rise to the next generation, right? So it's going to produce a seed. So seed is the basis for the sustainability of a plant generation. So this is going to have a short seed set. So now we are going to talk about geitonogamy. So transfer of pollen grains to the stigma of another flower. So this is in the same plant but between two flowers. So it functions as cross-pollination within the same plant. So it is genetically similar because the flowers are genetically similar because both the flowers are from the same parent. So the plant is the same. So genetically the gametes are similar. So last one is xenogamy. So it transfers from different plants. So this is essentially your cross-pollination. So they are genetically different types. So what are the agents of pollination? Now wherever, so there is cross-pollination happening in nature, right? So if we assure that cross-pollination is happening, so who are the agents that support cross-pollination? So who are the agents that help in pollination? So the agents are divided into two types. One is biotic agent, the other one is abiotic agent. So abiotic, that is a non-living agent. So the non-living agent here is wind and water. So wind pollination, water pollination, all right. The next one is biotic pollination. So biotic is living organisms. So here you have animals. So majority of the pollinators are animals. So to some extent you have wind and water. So Muhammad, hello, hi. Thank you so much for joining my session today. So please do like, share and subscribe. So I'm talking about the process of pollination in sexually reproducing plants. Right, so these are the two types of pollinators. So now for the pollen grains have to land on the stigma, and that is a chance factor. For example, if someone is going to pick you up and drop every day, say for example, okay, so my explanation is very good. The concept of geitonogamy, earlier I found it difficult, but now it's clear. So thank you, Rohan. So I'm happy that you have understood. Okay, so you are Safia Rafeen, right? Am I right? Okay, so hello and thank you so much for joining my session. So I am doing a class 12 topic today, right? So now, if someone is going to pick you up every day, suppose, say for example, you're going to go by auto to school every day. You are used to going by auto to school every day, and the automan is very important. So one day the automan is on leave, so then everything, so you automatically get tense because you have to find an alternate that day, right? So there are chances that sometimes you tend to miss something. So sometimes you get inside an auto on a normal day, but you forget something, but there's no way of turning back. But if it's your own vehicle, you can turn back, right? So suppose if you're dependent on someone, always you have to have an extra effort, right? So it is always good to be independent than to be dependent in a lot of cases. So suppose now the condition of the plants is that they are dependent on pollinators, right? Now, if they are dependent on wind pollination, so the wind carries the pollen grain, but there are high chances that due to some places the wind stops. Due to certain factors, the wind stops. Right? So there are possibility that the pollen grain is getting missed or it is lost, and the chance of pollination is very less, right? It's a chance factor. Ho sakta hai, it can happen. right? So just to prevent this failure probability, always your pollen grains are produced in a lot of number. So just like the human sperms. So human sperms are getting transported to the female vaginal tract, and to avoid the chance factor, they are always produced in large amount. So much in the same way, pollen grains are produced in the large amount, right? So to compensate, large number of pollen grains are produced. So first we are talking about wind pollination. So wind pollination is known as anemophily. So this is wind pollination, the other term for wind pollination, alright. So wind pollination is more common compared to water pollination. So we have two abiotic agents, one is wind pollination, one is water pollination, right? So among these two, wind pollination is pretty common compared to water pollination. So pollen grains here have to be light and non-sticky because it has to move along with the wind. So if the pollen grain is sticky, it will not be able to immediately fly along with the wind, right? So here the pollen grain should be light, it should not be very heavy, and it should be non-sticky so that it can be easily transported by wind currents. So here the stamen should be well exposed so that the pollen grains can be easily transferred. So now when the pollen grains from the exposed stamens, they are light, non-sticky, they can be easily passed by the air current. They should land on the stigma, and the stigma should also be receptive, right? So it should be widely open so that it can hold the pollen grain on itself. So the stigma should be feathery and it should be easily able to trap the pollen grains. Now how should the flower be? So the flower should have a single ovule. In each ovary, they should have numerous flowers. That is, it should be like an inflorescence, right? So that everywhere they can have the stigma and they are receptive to the pollen grain. So common example is seen in the corn cob. So they have tassels, that is the stigma and the stale. So it is also common in grasses. So in your corn cob, suppose, please bear with my diagram. So if this is your corn, so you have something like this. So that is your feathery stigma. So you can Google and find the picture. So sorry, I forgot to get the picture here. So this wind pollination is also common in grasses. But when we talk about water pollination is pretty rare. So it happens only in 30 genera of monocots. So genera is the plural of genus. So you have kingdom, phylum, class, order, family, genus and species, right? So genera is the plural for species, genus. So only 30 genus or genera of the monocots, they follow water pollination. So water is also the mode of transport in case of bryophytes, where that is the medium of fertilization, right? So example, Valisarnia and Hydrilla, they follow your water pollination. So let's see how, right? It's not that all the plants that live in water undergo water pollination, right? So just like we say, all the people who are good in biology are not that they love biology. They are good because it automatically comes to them, right? So much in the same way, when I say all the plants living in water do not undergo water pollination, examples are there. So like your water lily. So your water lily, it comes up to the surface of water, then it has a stalk and then the lily is there. So it is above the surface of water, right? So it undergoes wind pollination. So you have to remember this, not all aquatic plants or not all water plants undergo water pollination. So you have exceptions also. So people working in Nokia company might have an Apple phone. That is an exception. That's okay, right? So much in the same way. So examples here are marine sea grasses. Marine sea grasses are also known as Zostria. So not all aquatic plants use water pollination. So example, your water hyacinth and your water lily. So flowers emerge above the level of water, so they are pollinated by insects or wind. So if you can see, this is water lily and this is the water surface. So it is above the surface of water. So this is what I call it as a stalk, a filament-like structure. So now here is the flower. So here is my stamen or my pistil. So pollination happens by wind. Alright. So water pollination, another example, 𝗲𝗬𝗂𝗃𝗃. So this is a female flower. So it reaches the surface of water by a long stalk. The male flowers of the pollen grains are released on the surface of water. So water lily was an example to say that not all aquatic plants follow water pollination, but in 𝗲𝗬𝗂𝗃𝗃, water pollination happens. Fine. So male flowers of the pollen grains are released on the surface of water. So the currents, water currents, carry the pollen grains. So some male gametes eventually reach the surface of water and they are fertilized. So this is your 𝗲𝗬𝗂𝗃𝗃. Sorry, this is your zostrus, that is marine sea grasses, and this is your 𝗲𝗬𝗂𝗃𝗃. So the flower is above the surface of water. So further, this is above the surface of water, this is beneath the surface of water, both are 𝗲𝗬𝗂𝗃𝗃, right? So further sea grasses, the male flower is submerged inside the water. So in 𝗲𝗬𝗂𝗃𝗃, so the pollination happens above the surface or on the surface of water. But if you talk about your sea grasses, the plant itself is submerged underwater. Now how will the pollination happen? So the pollen grains are released inside the water. So here the pollen grains are long, ribbon-like. They are carried passively inside water to reach the stigma. So in water-pollinated species, the pollen grains are covered by a mucilaginous sheet to prevent it from getting wet. So if you're diving, so you wear clothes that make sure that you're not really wet, right? So you wear clothes of that material. So all the pollen grains that are pollinated by water, they have a mucilaginous covering. So both wind and water pollinated flowers, they do not have colorful petals or they do not have nectar. So nectar should be present when an insect is coming. So if you have noted, be it a wind pollinated flower, be it a water pollinated flower, they do not have the flower that is very bright and colorful, right? Yes. So now we go to animal pollination. So flowering plants are pollinated by animals. So the animals here includes your wasp, your flies, your bats, your butterflies, your moth, your birds, your beetles and ants. So pollination by bees is most dominant. So even larger animals, sometimes primates, the tree lizard, so primates, I mean the gorillas, the monkeys, they also act as pollinators. So animal pollinated flowers are very, very specific. Suppose a flower is being pollinated by bee, it has to be pollinated by bee. Next day, a wasp or a moth cannot pollinate the flower. So every flower is very fixed, it is very specific. Just like every substrate, every enzyme is specific to a substrate, every flower is specific to a pollinator. Alright? So insect pollinated flowers are large, they are colorful and they are fragment rich in nectar. So here the number of flowers are clustered and there are large number of flowers. The inflorescence is very, very significant. So conspicuous means significant, well developed. So animals are attracted to the color of fragrance. So if you note, I have told you, in wind and water pollinated flower, the petals are not very colorful. Right? Whereas in a flower that is to be pollinated by animals, the petals are very, very colorful. That is to attract the insects. So how will the insect come and help you? So it should be attractive and it should also provide some reward to the animal, right? So it provides nectar in return. That is why insect pollinated flowers have nectar and they are brightly colored. So flowers are pollinated by flies or beetles. They secrete foul odor and they attract them. Alright? So animal pollination, to sustain animal visits, flowers reward nectar and pollen grains, right? Hello Abhirami, yes, a very, very happy good evening. So thank you so much for joining my lecture today. So I am your favorite teacher. Wow, so you have watched my lectures before. So thank you so much Abhirami. So if you have any doubt in whatever I am teaching, so by chance you don't understand anything that I am teaching, so please do get back to me. And thank you so much for this compliment. So this compliment made my evening. Thank you. Right. So to give a reward, so it also produces nectar. But here, in case of wind pollinated plants, the pollen grain should be non-sticky. But here, it has to be sticky. Because the moment bee comes and sits on the flower, the pollen grain should be sticky enough that it sticks onto the surface of the bee, right? So that is how it is adapted, right? Yes, so here the pollen grain has to be sticky so that it can stick to the surface of the animal. Sometimes if the flower does not have nectar to offer, it will also offer a safe place for the animals to lay eggs. So sometimes laying eggs and protecting them from predators is a huge task, right? You would have seen birds also. So sometimes we humans themselves destroy their nest, and that's really sad. But still, sometimes the plants help the pollinators like this also. They provide a safe place to lay eggs as well. Fine. So example is Amorphous flower. So this Amorphous flower is huge. The flower itself is 6 feet tall, and the pollinator can safely go and lay eggs inside. So moth and the plant yucca. So moth is a type of insect and plant yucca is a plant. So they cannot complete their life cycle without each other. They are so close to each other that the pollinator of the yucca plant is the moth, and the moth has to lay eggs inside the plant itself, right? So the moth deposits egg in the locule. So locule is the cavity of the ovary, and the flower gets pollinated. So you can see the flower. So this is the huge flower, that is the Amorphous flower, alright, and this is the yucca and this is the moth. So how beautiful in nature, a lot of things live in synchronization, right? Fine. So now the next topic for this evening is outbreeding services. So sometimes you feel that you are too much good in your own world, so you have to deviate and you have to enjoy, right? Say for example, all of you are preparing for NEET, right? So you have been preparing for almost 6 months continuously without any break, without any distraction. Touch wood, you have done it well. Now you are saturated. Now you are tired. You are mentally tired of continuously studying. So you look for a distraction. So your friend says, come out, we'll hang out. So you'll have a good time, you'll have a good relaxation. Or say you're going out with your parents take you out for a dinner. So these are ways of relaxation, right? So this is also sometimes important. So continuous studying will also spoil your mental peace. You need to relax. You need to give time for yourselves to play with the competitive world, right? Fine. So something very similar happens in the life of the plant. Suppose if I have a plant which follows autogamy. So autogamy is self-pollination, right? So continuous self-pollination also saturates the plant. It is tired. So if you give me 10 cakes, I will eat. If you give me 100 cakes, yes, I am saturated, right? I cannot eat so much. So for next 3 months or 4 months, I will never talk about cake. So something like that. So continuous self-pollination also saturates the plant, and it causes the plant to have inbreeding depression. So it reduces the fertility level of the plant also. So now the plant will take a break, and the plant will prevent self-pollination and it will promote cross-pollination. Right? So now you have decided to relax, so you will close all your books, you will make up a plan that you come back in 2 days and then you study, right? So similarly, plants also take a break from self-pollination because that causes saturation to them, that also causes reduction in fertility rates, so thus they go for cross-pollination. But naturally the plant is designed for self-pollination. Now, if it has to prevent self-pollination and go against it and do cross-pollination, what are the adaptations the plant does? That is known as outbreeding services. Did you understand? Right? So, how does it do? So, majority of the flowering plants are hermaphrodite flowers. Hermaphrodite means the male and the female parts are present on the same flower. So, when the male portion and the female portion, so that is the stamen and the pistil, when they are present on the same flower, so that flower is called as hermaphrodite flower. It is also known as bisexual flower. Fine. So pollen grains and stigma are on the same flower now, right? So now continuous self-breeding leads to inbreeding depression. So flowering plants may have many devices to stop or suppress self-pollination and promote cross-pollination. So first they discourage self-pollination and they promote cross-pollination. But how does this happen? So pollen grains, so suppose if a plant is having self-pollination by nature, the pollen grains and the stigma, that is the male gamete and the female gamete, should mature at the same time. right? So that is when self-pollination will happen. But when the plant has decided to stop self-pollination, the pollen grains will mature at a different time, the female gamete will mature at a different time. So now obviously self-pollination cannot happen. So how smart is nature, right? So pollen grains released before stigma becomes receptive. So when the pollen grains become receptive or when they mature become the stigma, so that condition is known as proto-andry. So andrecium, andry is andrecium. So first the andrecium develops, that is proto-andry. So when the stigma matures before the pollen grain, that is also one method. Either the pollen grain can mature first or first the stigma can mature, right? So this is protogyny. So when the gynoecium matures first, this is protogyny. So Rohan is very excited to see you in CP Live lectures. Okay, I hope I am able to help you, Rohan. Right. So the pollen grains and stigma are placed in different positions. So when a plant is undergoing self-pollination, the pollen grain, that is the length of the stamen and the length of the pistil are of the same height. Okay. But when the plant has decided to stop self-pollination, the height differs. When they are in different height, then how will self-pollination happen? So this prevents autogamy, right? So they prevent inbreeding, self-incompatibility. So sometimes, suppose say I have two sunflower plants. Say I have two papaya plants, okay? I have a male flower and I have a female flower. So next to the female plant, I have another female plant of another flower, say marigold, okay? So now it is being pollinated by insect. But the insect, instead of putting the pollen grain or instead of landing on the papaya plant, it lands on the marigold plant by mistake. The pollen grain has fallen onto the stigma of the marigold plant. Now, do you think pollination, fertilization will happen? No. So the stigma knows which is the right pollen and which is not the right pollen. So the stigma has a level of interaction which rejects the wrong pollen grain. So sometimes to prevent self-pollination, if the pollen grain has landed on the stigma of the same flower, still the stigma rejects the pollen grain because it has to prevent self-pollination for its own good, right? Sorry, so this is genetic mechanism and this prevents self-pollination. So they inhibit pollen germination because once the pollen grain reaches on the stigma, so now it has to pollinate and reach the egg gamete, right? Yes. So further, another method is production of unisexual flowers. So bisexual flowers are never produced so that no pollination happens. So in case of castor and maize, so only one type of flower is produced. So then it is easy, right? So it is good to go for dioecious plant, right? So now the next topic is pollen-pistil interaction. So hi Veda, hello. So I guess this is Vedak. Okay, hello. So thank you so much for joining my lecture. So please do like, share and subscribe. So I will stop with pollen-pistil interaction because I've already told you. So the pollen grains reach the stigma. So how does the stigma know if it's the right pollen or wrong? Suppose instead of a papaya pollen, a rose pollen lands. So now it will not happen that way, right? A papaya flower cannot pollinate a rose pollen. It has to pollinate a papaya pollen only. So in that case, who's going to read? Who's going to do the cross check? Who's going to do the security check? That is the stigma itself. The stigma knows which is the right pollen. So stigma has the power to recognize it, right? So whether it is the right pollen or the wrong pollen. So if it's wrong, it is self-incompatible. So stigma has the ability to recognize the pollen. So if it's right or it is compatible. If it's the right pollen, it is compatible for fertilization. If it's a wrong pollen, it is incompatible. So when it's right and it's compatible, it accepts the pollen. The pollen germinates. So if it is a 2-cell stage pollen, it goes to form 3-cell stage and then it goes and fertilizes with the female gamete, right? But if it's wrong, the pollen grain will be rejected. So the stigma will not agree with the pollen grain. Yes, it is you. Amit Kumar, hello. So thank you so much for joining my lecture today. So please do like, share and subscribe. So the pollen deposition on the stigma until the pollen tubes enter the ovules is known as pollen-pistil interaction. It's a very dynamic process. So they are very quick in analyzing if it's the right pollen or the wrong pollen, right? So the pollen recognition is followed by production and inhibition of pollen, right? So we'll stop with this, and the next topic I'll continue in the next class because I think I have taught a lot of things. So we just have outbreeding services. So I will do outbreeding services or artificial hybridization later. So probably in the next class, that is on Monday. Monday we will almost talk about the process of double fertilization and what happens after fertilization. So we will stop here today, and I have an important announcement. So Monday, Wednesday and Friday I teach class 12, right? So Tuesday, Thursday, Friday I teach class 11. And in class 11 I am teaching plant physiology. So I am doing the first chapter in plant physiology, that is transport in plants. So transport in plants has a lot of problems as well. What is DPD? What is suction pressure? So what will be the DPD when the cell is turgid? So on and so forth. So tomorrow at 5:15 class, I am going to solve numericals or problems or application based questions from transport in plants. So please join me for the same so that you know how to apply the concept. Ma'am, when is the mentee going to be? So mentee will be there next week. Alright, Vedak? Yes? So Pakka, we'll have a mentee next week. Done? So please join me tomorrow at 5:15 because that session will be very, very helpful. I'm going to teach you how to solve problems, how to think. So that is one way I can help you. I can be of great help to you in solving numericals in biology. So please don't get scared. It is not really tough. It's going to be easy. I'm going to help you out. So don't miss tomorrow's session. So we'll continue with sexual reproduction in flowering plant on Monday. So until tomorrow 5:15. So please take care and have a great evening. Thank you and stay tuned. Bye.